Start with a thought experiment. Our everyday reality is just a surface layer. Beneath it, right under our own skin, operates a bustling landscape of cellular machinery that we are completely blind to. Our biology imposes a physical limit. Under 0.1 millimeters, the eye cannot separate two objects. They blur together. Magnifying that dot only creates a larger blur. Increasing resolution distinguishes detail, snapping the image into focus to reveal two distinct dots. To break past this anatomical blindfold, we had to engineer new tools. In the 17th century, Robert Hooke built early lenses, like this antique brass microscope, complete with a separate glass globe to concentrate light. This drawing from his book Micrographia illustrates exactly what Hooke saw when he examined a slice of cork. He observed these tiny, repeating hexagonal spaces, which he named cells. By bending light through curved glass, early scientists proved that a deeply complex, heavily populated universe exists right in front of us, just beyond the reach of human anatomy. The direct descendant of Hooke's design is the compound light microscope, the core tool of modern cellular biology, which relies on a stacked series of lenses to operate. This ray diagram illustrates exactly how that stack works. A condenser focuses the light, while the objective lens and the eyepiece capture that light and bend it, a process known as refraction, projecting a highly magnified virtual image into the viewer's eye. But a perfectly engineered optical machine is useless on its own. You cannot simply throw raw tissue under a lens and expect to see clear details. The chemistry of the specimen is just as crucial as the physics of the glass. For a quick look at a living, moving cell before it dries out, scientists use a temporary mount with a drop of water. To preserve a specimen for decades, they create a permanent mount, meticulously fixing, dehydrating, and staining the tissue to lock its internal structure in place. Modern preparation has evolved even further. Phase contrast techniques let researchers view living cells in their natural state without harsh chemicals. Fluorescence microscopy uses high-intensity light to make specific cellular targets glow brightly against a dark background, revealing hidden structures. Peering into the cellular world is a delicate partnership. It requires mastering optical physics to manipulate light and precise human chemistry to prepare the sample to receive it. Eventually, optical microscopes hit a physical wall. Visible light travels in relatively wide waves. When those waves try to pass through details smaller than 0.2 micrometers, they diffract and fail to resolve them. The wave itself is simply too big for the gap. To map the dense internal architecture of an organelle or catch sight of a virus, scientists had to abandon light entirely. Instead, they began shooting precise beams of electrons inside a vacuum. Electrons also behave like waves, but their wavelength is drastically shorter than visible light. This ultra-tight frequency allows the beam to effortlessly map incredibly fine details that visible light washes right over. This image of a paramecium was captured using a transmission electron microscope, or TEM. Firing electrons directly through an ultra-thin slice maps its internal architecture in two-dimensional detail. Alternatively, a scanning electron microscope bounces electrons off the surface, revealing a rich three-dimensional topography. By stepping outside the spectrum of visible light, the very medium our eyes evolved to process, scientists bypassed the ultimate optical barrier and began mapping reality at the nanometer scale. Modern microscopy requires a calculated trade-off. You can use light to watch a living organism swim and hunt, but you sacrifice ultimate detail. Or you can use electrons to achieve absolute resolution, but the harsh vacuum and metal coating required will kill the living specimen. Today, we continue to push these tools further. Digital microscopes, like the one shown here inspecting a microchip, output real-time imaging directly to screens, while super-resolution technologies squeeze past the traditional limits of optics. This ability to inspect the invisible underpins almost every sector of modern engineering. Manufacturers rely on these tools to verify the structural integrity of industrial plastics, metals, and the microscopic semiconductors inside our devices. In public health, they are essential for diagnosing malaria parasites in human blood. In the justice system, forensic scientists depend on them to analyze fibers, glass fragments, and ballistic evidence pulled from crime scenes. The history of microscopy is a continuous refusal to accept the biological blind spots we were born with, engineering the exact tools necessary to pull the deepest, invisible foundations of our world up into the light.